Okay, good afternoon, everybody, and welcome back to our discussion about DNA and DNA function. Remember that our discussion is largely slanted towards discussion about the prokaryotes. Although there's not a lot of difference, there are some differences between prokaryote and eukaryote function. But here's something that applies equally right across the board for all of evolutionary history, and that is that change in genetic material can and does occur. Although whenever a cell divides, the intent is that each succeeding generation should inherit all of the information contained in the DNA. If in fact we always handed on an identical copy, then evolution has nothing to work with, because evolution works on variability. If DNA replication, for example, was always 100% accurate, we would all still be swimming around in the primordial ooze. We would be prokaryotes. We would never have evolved beyond that stage if DNA never changed. But DNA does change. It can accumulate changes. And it does so continually, relatively slowly. Any permanent change in base sequence of the DNA is referred to as a mutation. And when a mutation occurs, it has only three possibilities. It either makes no difference, which can definitely be the case, as we'll hear, or the mutation can be beneficial, or it can be harmful. More frequently, it is harmful than beneficial. But beneficial change definitely does occur. And those beneficial changes are what fund evolution. Those beneficial changes improve the functioning of the organism, and the organism reproduces more. And when it reproduces, it hands on those beneficial changes to the progeny. So that is the stuff of evolution. The mutations can occur for various reasons. First of all, there are substances which increase the likelihood of mutations occurring, often because they affect the DNA directly or because they affect one of the repair mechanisms that the cell has. And cells have ways of checking DNA to try to eliminate mutations that occur. And mutagens, substances that increase the likelihood of a mutation occurring, work either directly on the DNA by causing mutations to occur, or else they work on the repair mechanisms and impair them so that mutations are not repaired by the normal cellular mechanisms. So spontaneous mutations occur in the absence of a mutagen. And they can be for many reasons. First of all, errors can be made during DNA replication. They can and are made during DNA replication. The DNA polymerases that are responsible for replication have proofreading ability. They can, as they do the replication, they can check to see that the correct bases are being laid down during replication. But they can occasionally miss. And replication is a major source of mutation. Mutations can also occur completely randomly through radiation, for example. We are bombarded by radiation the whole time, especially from the sun. And that in itself can be mutagenic. Okay, so we're going to look at various kinds of mutations. The simplest to understand is a single base mutation. One base in a DNA sequence is changed from one thing to another. That is called a point mutation or a base substitution. It's a change in one base in the DNA. Remember what an effect that can have, because this can change first of all, change a triplet in the DNA, in the transcript. it can, and that in itself may have an effect of changing a codon. It may change a codon from one amino acid to another. Let's see if we can find one. How about if we had a codon that read UUA, and the base in the triplet DNA was changed, so the codon now read UUC. You would have phenylalanine substituted for leucine. You get the idea. A single base change can actually have a big effect. And if you think back, I told you about the disease sickle cell anemia. That is exactly what happens in sickle cell anemia. A base is changed to substitute valine in the manufacture of hemoglobin, and it radically alters the functioning of the hemoglobin. Okay, so let's just have a look here, and we'll see the impact of some of these mutations varies as to where they occur, where and how they occur. So let's have a look here. And we're going to always consider this. This is the normal DNA. And here's the template strand of the DNA, organized, of course, into triplets here. And each triplet producing a codon on transcription, producing a codon in the messenger RNA. So here we're going to consider this triplet here, CCG, and we're going to change it to TCG. Okay, let's go back. CCG to TCG. That is going to produce codons that read GGC and AGC. What is GGC? GGC is for glycine. That's the amino acid glycine. Change this to T, and in the codon we have AGC, not GGC. AGC codes for serine. Completely different amino acid with completely different functioning. And a polypeptide with that substitution is likely to differ in its functioning. It may be beneficial, it may be deleterious, we don't know, but it is very likely that it's going to differ in its functioning. This is the normal. This is the mutation. So it may be very important. But notice something, and that is that the length of the polypeptide is not affected. The place of the next triplet codes for a stop codon. So the length and organization of the gene has not been changed at all. It's just the base. A single point mutation has resulted in the substitution of one amino acid for another. So let's have a look at this one. And this here again, here's the template here. Here's the messenger RNA strand here. And there's a methionine there. We're looking now at this gene, at this triplet here, TTC in the DNA. That's the normal. But if we have a base substitution of A for the T, which is actually very easy, one of the easier mutations to acquire, here we have ATC instead of TTC. And ATC doesn't actually code for an amino acid, it codes for a stop codon. This here is the normal length of the transcript. Here is the normal length that would be translated. But now we've introduced, right down here, we've introduced a stop codon by mutation. And this is called a nonsense mutation, because what it does is it introduces an inappropriate stop. The translation will end before the polypeptide is complete. And in all likelihood, it's going to be completely non-functional. Okay. So nonsense mutations can be extremely deleterious There's very seldom that they would be beneficial. So this is each of those. The structure of the triplets and the structure of the codons remain the same. You'll see what I mean now. This kind of mutation that I'm going to discuss now is called a frame shift mutation. And look what happens. Here we are. We're going to look at this here, this triplet here, AAA, and its codon will be UUU, which codes for phenylalanine. But now what we're going to do is remove one of the A's. Take one base out. And what happens? What happens downstream? Well, we now have completely new triplets. We've taken out a base. We now introduce entirely new triplets. We have to restructure everything downstream. Where we've taken one out, we have to restructure everything downstream. And that all of those changes will continue until such time as we somehow, if we were to be able to put in another base and restore the structure of the triplets and restore the structure of the codons. So this is very, 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 very damaging, extremely damaging, because what you're in fact doing is you create, just by taking out that one base, you're creating an entirely new DNA sequence. This sequence here, our normal, is methionine, lysine, phenylalanine, glycine, and then the polypeptide, then translation should stop. Here, look, up till that, where we took that base out, it says same methionine, lysine, but now the new DNA sequence says leucine, alanine, and on and on and on and on. We don't know where a stop codon is. Frame shift mutations are extremely deleterious because they restructure the entire DNA sequence downstream of where they occurred. If, just think for a minute, if I was to take out two bases instead of just one, I would have a completely different DNA sequence to what I have here, but it would have the net effect would be the same. I would restructure the complete DNA sequence downstream of that removal. How about if I take out three? How about if I take out an entire triplet and thus an entire codon is missed in translation? We can't predict what the effect would be, whether it would be beneficial or deleterious, but what it would do, if I took out three, it would remove an amino acid from the polypeptide, but it would not change the downstream sequence at all. So if I take out one or two, radically change the downstream sequence. Take out three, you remove an amino acid, but you don't change the downstream sequence. Why? Because everything is in units of three. Okay. A similar situation will arise if I was to put in a base. And that can happen. A base can be inserted into a sequence, and then it radically changes. It changes the sequence downstream. If I put in one or I put in two, I restructure the entire downstream sequence. If I put in three, I put in a new amino acid, but I don't change the downstream sequence. Frame shift mutations are extremely dangerous, and it can have radical effects on the DNA sequence and on the outcome of translation. Okay, so we are now going to go on to just talk a bit about prokaryote genetics in general, about some aspects of prokaryote genetics. But I'm going to then move on to a very, very brief discussion about genetic engineering. And I don't have the time to go into it in detail. If you are interested in it, then a course in cellular biology is what you want to do. But I will just introduce some of the ideas to you so that you have an idea of how and why bacteria, for example, are such a favorite subject for genetic engineering. The first thing we need to talk about is that the bacteria, that prokaryotes, often have extremely mobile genetic elements in the cell. And that is that they can move small parts of their genome, let's say, around their own genome, or they can move it from one cell to another. It's an extraordinary thing. There are a few eukaryote organisms where there is this kind of mobility also exists, but they are few and far between compared to its frequency in the prokaryotes. The first one I have already mentioned in previous classes, and one of the most important for us to understand amongst the mobile elements are the plasmids. Plasmids look like miniature chromosomes. They are small circular pieces of DNA, and they are self-replicating. Sometimes plasmids replicate at the same time as the rest of the cell replicates. Other times they do not. They replicate independently. Depends on the plasmid. And they can be quite large, and they can contribute a considerable amount to the whole genome. The bacteria, they're up to 5% size of the bacterial chromosome, and some cells have none, but many prokaryote cells carry a population of these plasmids. And there are a number of things to say about the plasmids. First of all, they are infectious. That is, they can move. If you have plasmid which is free in the environment, it can actually be taken up by living cells. It's a very important aspect of prokaryotic ecology. The second thing is the plasmids often code for proteins that are very advantageous to the cell. They code for all sorts of things, but for example, they can confer on a cell resistance to things like heavy metals, resistance to antibiotics. They can also confer things like virulence factor. They can enhance a cell's ability to be pathogenic. And all of these things are advantageous to a prokaryote. So here, for example, this is called an R. This is called the R factor. I'm just using it as a classic example of a plasmid, and I'll explain exactly that this plasmid has a peculiar way of being transmitted from one cell to another. So here it is. Here you can see small circles of DNA, in this case much smaller than the chromosome. But if we look at the DNA sequence, we can actually identify the different things about this plasmid and why, in fact, it will become so advantageous for a cell to acquire it. First of all, here's a gene here on the plasmid for mercury resistance. Mercury under normal circumstances is very toxic, even to bacteria, but this gene confers resistance to mercury. Next, it confers resistance to many different antibiotics. Here they are: sulfonamide here, streptomycin here, chloramphenicol here. These are all fierce antibiotics, by the way, and tetracycline here. So all of this here is almost half of the plasmid is devoted to conferring resistance. The other half of the plasmid is devoted to enabling a cell which has this plasmid to actually conjugate with another cell. This is kind of like bacterial sex. It's like bacterial sexual reproduction in a way, because what is happening here is this allows a cell with this plasmid can get together with another cell and can actually inject a copy of this plasmid into the other cell. And that indeed does happen. We'll talk about it more in a while. But realize that when that happens, when these cells actually conjugate and exchange DNA like that, this plasmid, a copy of this plasmid is transmitted, and along with it, all of these resistances. So now I'll just introduce the idea right now that if we were able to extract plasmids like this, purify them, and then manipulate them, maybe cut them and put in genes of our own desiring, and then get them into living cells, we could actually completely transform that living cell. We could change its genetics entirely by introducing new genes. And this indeed is what we do in genetic engineering. Very often we will use plasmids. They are a very frequent vector. That is the word for them. They're a frequent vector for genes which are of interest to us. Maybe getting a bacterium to start producing a new protein. Maybe getting a bacterium to manufacture insulin for us, or something like that. This can all be done using a plasmid like this. And not only that, if you use a conjugation plasmid like this, where it allows a cell that successfully takes this up to start conjugating with others, your desirable gene very swiftly can get spread through an entire bacterial population. Okay, so there are different kinds of mobile genetic elements. The plasmids are only one of them. But there are different kinds of plasmids. I just mentioned that one we just discussed is a conjugative plasmid. It carries genes for conjugation. It is also a resistance plasmid because it encoded antibiotic resistance. There are very frequently plasmids may also carry genes which allow a prokaryote that receives them to begin utilizing a new substrate and to catabolize a new sugar, for example, that under normal circumstances the prokaryote could not do. A typical example would be lactose. Many bacteria cannot ferment lactose, but we can introduce genes to them which allow them to use lactose, and we indeed do that. We often do it as a way of marking prokaryotes that we have successfully changed by genetic engineering. We'll see that in a while. Very often plasmids carry virulence factors, and virulence factors may be various things. They could be coding for something that acts as a toxin. That's one thing. They could also be coding for something that allows a cell to become pathogenic, for instance, by evading the immune system. And we're going to see one very important example of this in a while. Lastly, we're not going to talk about them much. A very fascinating group of mobile genetic elements called the transposons. Now, transposons are not plasmids. They are usually part of the bacterial chromosome itself. But they are very mobile within the genome. They can move from one place in the genome to another, and that can have various effects on the functioning of the cell. They also can be exchanged. They can move from cell to cell. The transposons, I'm not going to discuss them in a lot of detail, but we believe that many of the transposons originate from viral infections. And so and they can be transmitted by viral infection. Okay, so here it comes one of the most important experiments that was ever done in microbiology. This experiment actually opened up, even though they didn't realize it at the time, this experiment opened up the whole field of genetic engineering. It sits as the cornerstone, excuse me. It sits as the cornerstone of the whole science of genetic engineering for a reason that you'll appreciate in a minute. This is an experiment done by a scientist called Griffith, D.W. Griffith. And he was working on Streptococcus pneumoniae. The Streptococcus pneumoniae, it's a typical Streptococcus, but it consists of two cells, two coccoid cells, which stick together. So it's a diplococcus. And it causes a very, especially in elderly people or in people who are debilitated, it causes a very virulent pneumonia. It used to often be called hospital pneumonia. And it is the kind of pneumonia which very frequently ends the life of an elderly person. So it's always been recognized as a very important disease, and Griffith worked on this trying to develop a vaccine. By the way, there is now a very good vaccine against this, which you should all eventually have. So he was trying to develop a vaccine, and in doing so, grew up masses and masses of Streptococcus pneumoniae. It's very easy to grow on medium. And he realized that there were two strains of the bacterium. The first strain, the cells look identical, excepting they have a big, thick capsule around them. So here's two examples of big, thick capsules. And this is the virulent form. It's also called the smooth form, because when you plate it, as I say, it's very easy to grow. When you plate it, it makes colonies which are glistening and kind of mucoid, slimy, and shiny. So this was designated smooth. And if you inject the smooth bacterium into a mouse, the mouse dies of the disease. Then the other variety that he found were these. The cells look identical to these, Diplococcus, but there's no capsule around them. And if you plate them, they make a distinctive, rough, dry-looking colony. And that is because they lack this capsule. It's all the accumulated capsule that makes this slimy and shiny. In the rough, there is no capsule. So it has a dry, crusted look to it, and it was called rough. And if you make a suspension of rough and you inject it into mouse, the mouse survives. So the next steps need to be watched very carefully, because they look simple, but they're actually quite profound. First of all, he found that he could heat kill smooth. So if he heated it up, pasteurized it effectively, he could kill it, and he could inject it into mice, and nothing happened. The mice did not die. They stayed alive. But he couldn't plate any of them because they were dead. So they're not going to form colonies at all. Here comes the crunch. What he did was he took rough, a suspension of rough, and he mixed it with the same suspension of heat-killed smooth. And he injected it into mice. And guess what happened? The mice died. This is very strange, because what he's doing is he's injecting live non-lethal rough, and dead smooth. And this was a mystery until he actually extracted bacteria from the dead animals and plated them. And what did he find? He found that they grew smooth colonies. What has happened? There's no living smooth. There's no living smooth. He's proved that. There's no living smooth at all in this suspension. When he mixes this suspension with living rough, not only does the mouse die, but the bacteria change. The rough bacteria are transformed. That is a real word. It's not a transformation now acquires a new meaning. It's not just that they've changed, it's that they have changed for a specific reason. And the reason is that these rough cells have acquired the genetic information to create capsule. If you're able to look at this suspension of dead cells, they're not just floating around like this. In the dead pasteurized solution, there were many, many, many broken cells. And the suspension contained not only whole cells, broken cells, slime from the capsules, etc., etc. It also contained free DNA. And the DNA had on it the information to create capsule. And that DNA was taken up from solution by these bacteria, and they were transformed. When we talk about transformation in microbiology, we specifically mean that a cell has acquired DNA from another source. The cell has been transformed from one genome to another. Its genome has been altered by the acquisition of genetic information from another source. What is extraordinary about this is that Griffith knew nothing about DNA. This was in the 1920s. This was 35 years or something before the structure of DNA was elucidated. But he realized the real significance of this, that in fact we could, by mixing hereditary material with living cells, we could actually transform cells from one type to another. What they've acquired, in actual fact, is a relatively simple thing, and it's the gene to create the capsule. Because what is happening is in the smooth bacteria, the capsule allows the bacteria to evade the immune response, and it allows cells to attach to respiratory surfaces, for example, to grow. So the capsule is really important in the pathogenicity of this organism. It can't, the immune system of the mouse, for example, its macrophages can't ingest the encapsulated bacteria. So they evade that response, but they can also attach much better. Rough, they are very easily eaten by macrophages and very easily dealt with by the mouse's immune system. So it does not get sick. This is virulent and it dies. Okay, now I'm going to run this. I'm just going to try one thing because I know that there have been problems with people hearing. Let me see if I can just do this. I'm just going to briefly do this. Okay, let me just check here. I hope that worked. If it didn't, if you didn't hear the sound, you will be able to hear it when you refer to the PowerPoint that I posted on Canvas. Okay, so here's a point to emphasize. I did mention it, but I'll re-emphasize. After he had done this whole experiment and he had mice that died from transformed bacteria, he was able to extract bacteria from them, and they were smooth. They were smooth bacteria. They had acquired a gene. Okay, so a couple of the things I'm going to talk about now are of particular importance when we start to think about genetic engineering. In genetic engineering, very often what we want to do is we want to take genes that we are interested in and we want to put them into bacteria. So the first thing to say is bacteria in the environment actually can very often take up DNA, free DNA from the environment. Those bacteria that are able to do so, we call competent cells. Not all bacteria can do this. And for example, E. coli doesn't do it very easily. It will do it by the method that I'm going to show you now, by conjugation. But in general, when we're working with E. coli, which is our commonest experimental organism in genetic engineering, we have to make the cells competent. They are not naturally competent. They won't naturally take up a plasmid that we've constructed, for example. Instead, we have to do something. And often it's very simple. Sometimes it's as simple as heat shocking, a brief pulse of heat to the cells. Sometimes we can do it with a brief electrical pulse. And what that happens is that somehow the membrane is affected so that it becomes permeable to the DNA, and the DNA can be taken up from the environment. This, however, is another system. This is a system I have already mentioned. We saw this plasmid a little while ago and said that this was a conjugation plasmid. And the whole plasmid is referred to as the F factor, fertility factor. And cells which carry it are called F+ cells. And this plasmid confers various things. We saw that it conferred mercury resistance, antibiotic resistance, et cetera, et cetera, et cetera. But it also confers the ability to conjugate, to transfer genetic information from one cell to another. This is a natural system. In populations of naturally occurring bacteria, this is going on the whole time. We even discovered it. And all the time I'm talking about it, just think how useful this system is if we want to insert a gene of interest into bacteria which do not normally have that gene. So for example, maybe I want to put in the gene for insulin, and I put it in here somewhere. And I now take, if I can now get this plasmid into a cell, I will transform that cell. And I'll transform that cell into a cell which can conjugate. I'll transform it into an F+ cell. And I can then have that F+ cell distribute my gene of interest through a whole population of bacteria. And here's how it works. F+ cells are able to make a thick tube called a sex pilus. It's a proteinaceous extension of the cell, and it will join up to a neighboring cell, which is F-. Sorry, it's this way around. This is the F+. This is the F- cell here. Here's the sex pilus, and the sex pilus has two functions. First of all, it attaches, but then it shortens, and it draws the cells very close together, and it widens into this bridge here. It's called the mating bridge between the cells. It's not entirely clear what is going on here. Some people believe that the cell membranes actually unite here, others that there's still a protein bridge kept. But either way, a passage is created for DNA to pass from this cell into this cell here. And what the donor cell, this F+ cell, does is it reproduces that plasmid. Here it is here. So here the mating bridge has been established. Here it replicates that plasmid and it feeds it into the cell here. And that then heals up as an entirely new plasmid. And what has happened now is that you have seen the transformation of an F- cell into an F+ cell. And in addition, the F- cell has acquired all of the genes that were present on that plasmid. And as you saw, they were considerable. This cell has now acquired all of those resistances, et cetera, and is now functionally acquired those to then go on and conjugate with other cells and transmit it. There's something really important to notice, and that is that this chromosome here can be entirely different to this. These two cells can be from completely different strains with an entirely different DNA sequence on the chromosomes. It's as long as they are compatible for conjugation. Conjugation can occur, and this plasmid can be copied into. On this side here, it replicates everything again. This side here, they double up their DNA, et cetera, and they, as far as the plasmid function goes, they are identical. So there's just a little sort of a twist to this, and that is that sometimes that plasmid that's injected during conjugation can actually integrate into the bacterial chromosome. And when this occurs, it confers the ability for this cell to conjugate with very, very high frequency. They just do it the whole time. In genetic engineering, it's very useful because what happens is that this plasmid now does not, it breaks and it reproduces part of the plasmid, and then part of the chromosome is injected into the cells that they are conjugating with. They do not inject the whole plasmid usually, because the plasmid, the F factor rather, which was the plasmid, it breaks somewhere here. And then it reads off a whole lot of the plasmid and then of the bacterial chromosome instead of reading across just the plasmid. And this is important because this means that it does not, you can see here, it breaks. We get a piece of the plasmid and then we get a piece of the chromosome being transferred across. And that now integrates into the cell, into its chromosome, and it is carried across. It doesn't carry the entire plasmid. It does not allow this cell now to become F+. It remains an F- cell, but who knows what genes did it acquire from the donor cell. It not only might have acquired some useful genes on the plasmid, some of those resistance factors may have gone across, but it's also acquired genetic information from the chromosome of the donor cell. And that may be extremely useful. This may also provide us with a way of actually transforming the chromosome, the entire chromosome of a recipient cell. And this is a useful technique in genetic engineering. We may use this deliberately to transfer genes from one set of cells to another without making them F+. cells. So that's another use of conjugation. Let's see if this works. Okay, and this is an explanation of HFR stands for high frequency. Oh, sorry. See, I can. This is actually not that important. That will be on your PowerPoint set, so you can use that for revision. Okay, so that's one way. Okay, we can transfer. We've seen in Griffith's experiment, we saw transformation, taking up of free DNA from the environment. In conjugation, we saw how DNA can be transferred from one cell to another cell by conjugation. Here's a third way in which bacteria can acquire DNA, and that is by transduction by viruses. Now, this is the normal functioning of a virus. Here's this beautiful thing here is a bacterial virus, a phage, a bacteriophage, and its normal functioning is to find bacterial cells to which it can attach successfully, and then to inject its DNA into the cell. So what's important about this is this elaborate structure here remains outside, and it injects naked DNA into the cell, which may have various fates, but the intent is usually that this is viral DNA, and it contains all of the genes for manufacturing the whole virus. Okay. When that virus is busy being made inside the bacterial cell, it happens mechanically that manufactures all of these proteins that self-assemble into this elaborate structure inside the cell. The new viral particles are made inside the cell. And once they are made, then copies of the DNA are packaged into this head. And then the cell bursts and it releases the particles to go out and infect new bacterial cells. It happens naturally that sometimes the phage particles will package bits of DNA that are not viral. But all of the infectivity of the virus is here. It's all in this particle here. It's all in this elaborate protein structure. And no matter what DNA it has inside it, if this is all correctly structured, this particle can go and attach to bacteria and inject, and it'll inject whatever DNA it has in it. The normal is, of course, that it's injecting viral DNA, but we can set up a system where we exploit this, and we get prokaryotes infected with a virus, and instead of packaging viral DNA, we get the viral particles to package DNA which is of interest to us, and we then let those viruses go and infect bacteria and inject into them DNA, which is of our construction or our interest. And that is the system of transduction. Transduction is transformation of bacteria by having viruses inject the DNA. And it's a very, very useful system and is widely used in genetic engineering. So we need to ask the question, why on earth what is biotechnology? Why do we want to do it? Well, biotechnology is the use of microorganisms, not only bacteria, but any microorganisms, yeasts, cells, whatever, to make products which are useful to us. And they're now used extremely widely to produce food components, for example, to produce antibiotics, to produce vitamins, very, very importantly, to produce enzymes, and also to do things like produce hormones such as insulin, that sort of thing. The reason that we're so interested is, in fact, because bacteria are, in fact, very, very easy to grow. It's very easy to get them to grow in huge populations, even if each individual cell, bacterial cell, only produces a small amount of the product we're interested in. It is possible to set up huge cultures of them, millions of liters of culture, and have them actually produce an enormous amount simply because it is so easy to get their numbers up to very, very high levels. So what the whole system of biotechnology basically depends on is on us being able to take DNA and construct DNA of interest to ourselves by various means. We manipulate DNA and we get our DNA of interest to recombine with naturally occurring DNA. We allow the normal functioning of cells to continue because they have their own DNA, but we recombine DNA of our interest into their genome. That is why we refer to it as recombinant DNA technology. We get DNA which of interest to us or of use to us or whatever, and get it to become part of the genome of a host cell that we have selected, which we know how to grow and everything else. So first of all, just some to remind you of, you've heard these words before, but a vector is a DNA molecule which is used to transport foreign DNA into a cell, and usually it is self-replicating. So the plasmids, just like the chromosome of prokaryotes, the plasmids usually have an origin of replication. They may be, that replication of the plasmid may be tied to replication in the cell itself, but very often plasmids operate independently, and in fact we can manipulate them, add genes to them, which ensure that they make multiple copies of themselves within a cell, so that we get lots and lots of activity around the genes that we've attached to those plasmids. I remind you also that if I take a single cell with a particular genome and I allow that cell to reproduce over and over and over again, what I've done is I've created a population of clones. Clone is a population of genetically identical cells which have arisen from one cell, and they have an identical genome. If they had a particular chromosome, the chromosome is identical through all of them. If they had plasmids, the plasmids are identical through all of them. And so this idea of growing up clones is very useful because it means that we get a large population, which can be subsampled, which all carry exactly the same properties. Okay, so here is just a typical kind of system of genetic engineering. First of all, we have a cell which has a plasmid, and that plasmid we probably well characterized plasmid. Many of the plasmids are well known, and we understand exactly what its size is and everything else. We grow up a huge population of these bacteria, and we then extract the plasmid. We would kill the cells, lyse them, extract all of the DNA, and then we would sort the DNA. Usually what we would do is we would sort it by size, because we know pretty well what the plasmid is, what its size is. In addition, I should tell you, we usually make sure that we include, if we're going to insert anything into this plasmid, we make sure we insert a marker that will allow us to see that, in fact, our DNA of interest has gone into the plasmid and that it is operating. And we'll talk about that again in a little while. So we have our plasmid, and now we have a source of DNA, maybe from the chromosome of another bacterium. Maybe this chromosome has a gene to do something which we think is useful, which this plasmid doesn't have. So we can cut the DNA, isolate. This sounds simple. I sound simple the way I'm doing it, but this may be the most difficult part of all. We isolate the gene that is of particular interest. We'll leave it at that for the moment. And then we insert it into the plasmid by means that I'll explain briefly later on. And we then render a cell competent to take up the plasmid. We let the cell take up our new recombinant plasmid, and we then look for the activity that we're interested in. I mean, there are just a plethora of different things that we could use it for. We could use it, for example, to actually influence the genetic behavior of other things like plants or whatever. We could be using it, we could have inserted into that plasmid one of those enzymes, the genes for an enzyme which can break down stuff, detoxify, whatever. We can use it to produce useful enzymes, amylase, cellulase, all sorts of things. We can produce product from these genes. And then, as I've mentioned before, these now are very rich sources of hormones, which before were very difficult to extract. For example, insulin was usually extracted from pig pancreas. A very difficult process, a very laborious process, but pig insulin is identical to human insulin. So it was actually extracted from living animals. Nowadays, almost all of our insulin is produced by recombinant DNA technology. In addition, there are other hormones like human growth hormone or things like that that are also produced by recombinant techniques. So it's a very, very useful procedure. Let's see if this will make it clearer. Okay, so there is this very neat trick, and it is to use these enzymes referred to as restriction enzymes. Restriction enzymes are naturally occurring enzymes in prokaryotes, and their actual purpose is to break up viral nucleic acids. And to do so, each restriction enzyme responds to a very particular sequence of bases. In this case, for example, this restriction enzyme is recognizing this sequence, GAATTC. Here it is on this side, GAATTC, something important. This is called a palindrome. It reads the same forwards as it reads backwards. GAATTC, GAATTC. It recognizes that sequence, but it cuts it here between the G and the A there. And it produces this step in the DNA, which we call you heard it referred to there as a sticky end. So here it cuts here and cuts here. In between here is our gene of interest. Now that's the difficulty. This is the difficulty, which I'm not going to go into a huge amount of detail about because it's the subject of lecture after lecture, how to actually find and isolate our gene and identify a useful site like this on either side of it. That's what you eventually you have to do in order to extract a gene of interest. That could be many months, many years of work to actually do that. But this is the end result. We identify a gene of interest, and on either side, we identify a restriction site that we can cut. Here it comes here. It produces this sequence AAATT, and here's the C and G. Here AATT, and here's the C and G. So it has a 1, 2, 3, 4 base sticky end. So this is the trick. We cut with this restriction enzyme. It cuts exactly there, right? If we now go to our plasmid and we mix plasmid with that restriction enzyme, the restriction enzyme will cut at exactly the same place, and the sticky ends will match. We will be able to take our fragments from another source, and we will be able to insert it into the plasmid. Such a neat trick, simply because the restriction enzyme cuts at exactly the same sequence and produces exactly the same sticky ends and the plasmid as it did on our source DNA. And that allows us to produce this recombinant plasmid. We had an original plasmid, and we have recombined it here with the DNA, with our DNA, with a DNA of choice that we are interested in. And thereafter, the plasmid should function perfectly well, provided we haven't inserted that gene into something essential, like the ORI or something like that. Okay, so this is typical. Here is a typical plasmid used for cloning. It has its own ORI for replication, and it has a very important gene on it, and that is this gene here. It's called the LAC-Z gene. And the LAC-Z gene is a gene which confers the ability to ferment lactose. It also has on it a resistance plasmid, ampicillin there. And here in the LAC-Z gene, it has 3 sites, 3 restriction sites, very well-known ones. This is called Hindi3, BAMH1, and ECHO-R1. And this plasmid is a very famous one. It's called PUC19. Very, very, very commonly used for doing for recombinant work. For this reason, first of all, it has an ampicillin resistance gene on it. So if we insert this plasmid into cells which do not naturally have ampicillin resistance, we will confer ampicillin resistance on them. So only those cells which have required acquired, our plasmid, will grow on media containing ampicillin. It's a way of selecting. We select the cells which have been successfully transformed with our plasmid by growing them on media which contain a antibiotic to which they are resistant, in this case ampicillin. But there's another one, and I think I'll leave the entire explanation of it till next time. I'll just leave you with this. This LAC-Z gene here is for fermenting lactose, and there are ways we can tell on our media whether cells are fermenting lactose or are not fermenting lactose. And if we insert something into the, if we cut this with Hindi-3 restriction enzyme, we end up with cutting this here in the middle of this LAC-Z gene, and we then insert into that Hindi-3 site DNA which is of interest to us. What we'll have done is we will have disrupted this gene. The cell will no longer be able to ferment lactose. So now we have two things. We can tell, did a cell acquire our plasmid? We tell that by seeing whether it is resistant to ampicillin. If it did, then it received the plasmid. But that's not good enough, because we will also be able to insert into those cells PUC19, which don't carry our useful gene, right? But if we select those which grow on ampicillin and which no longer ferment lactose, because the lactose gene has been disrupted by us inserting something here, then we have selected those which we have successfully transformed with recombinant DNA. So I'll leave it there, and then we can we'll go over this again when we start our next class.